You are carrying DNA from a species that no longer exists. That is not a theory. That is not a fringe claim. That is the consensus of modern genetics. Approximately 1 to 2% of you, if you are from a population outside sub-Saharan Africa, comes from Neanderthals. Some populations carry an additional component from Denisovans, a species we know almost entirely because their DNA appears in living humans. Their physical remains amount to a finger bone and 2 teeth. The species exists in the scientific record primarily as a signal in you. And there are others, populations with no bones at all, no fossils, no artifacts, known only as a sequence in living people. Geneticists have a term for them: ghost populations. The Sumerians wrote 6,000 years before genome sequencing existed that humans were made by mixing clay with the body of a killed being. They said that being's Etemmu, its permanent residuum, its non-physical component, was integrated into every human made through that procedure. Etemmu translates almost exactly as ghost. This investigation is a collision between what a laboratory found in 2010 and what a scribe wrote in 1700 BCE. In 2010, Svante Pääbo and his team at the Max Planck Institute for Evolutionary Anthropology published the first high-coverage sequence of the Neanderthal genome. The findings were published in Science. The finding that changed the field. Modern humans outside of sub-Saharan Africa carry Neanderthal DNA, not as contamination, not as error, as inheritance. Genetic material passed down from ancestors who shared territory with Neanderthals and reproduced with them. The sequences are integrated into the living human genome the way any inherited trait is integrated. They are expressed in measurable ways. Variants of Neanderthal origin influence immune function, metabolic processing, skin and hair characteristics, and neurological traits in the populations that carry them. Pääbo received the Nobel Prize in Physiology or Medicine in 2022. The finding is not contested. The percentage is approximately 1 to 2% across populations outside sub-Saharan Africa. It is not the same in every population. Different groups carry different amounts, reflecting different histories of contact and admixture. But the pattern holds universally. No population outside Africa has 0 Neanderthal ancestry. The Neanderthal sequences in modern genomes are not inert. They are expressed. Studies conducted after the initial genome sequencing identified specific Neanderthal-derived variants associated with immune system function, the innate immune pathways that respond to pathogens. Some of these variants appear to have been positively selected in populations that moved into regions where Neanderthals had lived. The Neanderthal immune experience, accumulated over hundreds of thousands of years in environments that modern humans were entering for the first time, was useful. It was kept. Other Neanderthal-derived variants are associated with metabolic function, with pain sensitivity thresholds, with the regulation of certain neurological processes. The inherited sequences are not a museum exhibit in the genome. They are active participants in how the body that carries them operates. The technical term for the process that put them there is introgression, the incorporation of genetic material from one species into another through hybridization. When two populations that are genetically distinct but reproductively compatible share territory over a sustained period, genetic material crosses the boundary. The offspring carry sequences from both lineages. The sequences persist through subsequent generations, even after contact between the populations ends. Introgression is a normal evolutionary mechanism. It has been documented across hundreds of species. What was not expected, what the pre-2010 consensus explicitly predicted would not be found, was that it had occurred between Homo sapiens and archaic human species at a scale detectable in every living human population outside one continent. The clean lineage model of human evolution, one species emerging, displacing all others with no reproductive crossing, is no longer the model. It has not been the model since 2010. What we are is a mixture. The cleaner question is, a mixture of what exactly? And how much of it are we still processing?
The Denisovans are the most unsettling case. When the Max Planck team sequenced the DNA from a fragment of finger bone recovered from Denisova cave in the Altai mountains of Siberia, they found they were looking at a population genetically distinct from both modern humans and Neanderthals. A third lineage. A separate branch of the human family tree that had been living and dying and moving across Asia for tens of thousands of years. Their total known physical remains: one finger bone, 2 teeth, a fragment of jaw recovered from Tibet. That is the entire Denisovan fossil record. What we know about Denisovans, their geographic range, their interaction with other human populations, their contribution to living humans, comes almost entirely from their DNA signature in the people who descended from them. Modern populations in Oceania, Southeast Asia, and parts of East Asia carry Denisovan sequences at elevated levels. The contribution is detectable, measurable, functionally significant. A species that we know almost exclusively as a code in the living. This is the concept the field calls a ghost lineage, a population whose physical existence has left no accessible trace in the fossil record, but whose genetic contribution persists in living descendants. It is inferred from the signal, not from the bone. There are others. Analysis of admixture patterns in various populations has identified contributions from populations whose physical remains have not been found and may never be found. The statistical signatures are clear. The sources are unidentified. They are logged in the genetic literature as uncharacterized archaic admixture, sequences that do not match any known species but are detectably not from anatomically modern Homo sapiens. Ghost populations, known only by their persistence in you. Stop on that for a moment. A species existed. It lived somewhere. It reproduced. It died over tens of thousands of years. At some point, individuals from that species and individuals from the population that would eventually become modern humans shared territory and reproduced. The resulting offspring carried sequences from both. Those sequences were passed down. They are in living people today. No bone, no tooth, no artifact, no cave painting, no campfire. Not a single physical trace of this species has been recovered from the ground. What we know about it, that it existed, roughly when, roughly where, roughly what its relationship was to the modern human lineage, we know because of what it left inside the people who came after it. It exists only as a pattern in the living. That is what ghost means in this context. Not absent, invisible except as a trace in something that persists. And there is one more category that belongs in any serious treatment of how much of you is not straightforwardly human. Approximately 8% of the human genome consists of endogenous retroviruses, versus viral DNA, that integrated into ancestral genomes over millions of years and became heritable. You carry the genetic remnants of infections that happened to organisms who lived before you. Some of these sequences have been co-opted for essential functions. The syncytin proteins involved in placental development come from ERV sequences. The mechanism your body uses to grow the tissue that sustains you in the womb was borrowed from a virus. 1 to 2% Neanderthal, some Denisovan, uncharacterized archaic sequences from ghost populations, 8% viral. The percentage of you that is straightforwardly, purely Homo sapiens is in the low 80s at best, and that estimate uses only the categories genetics can currently identify. The Atrahasis epic is a Babylonian text whose principal fragments are held in the British Museum. It is the creation account this investigation has examined twice before. Its relevant passage describes the creation of the first humans with a precision that becomes more interesting the more carefully it is read. The procedure has 3 components: The first is clay, Ṭēmu in Akkadian, the physical substrate, the base material. The second is the biological contribution from we-ila, the member of the prior workforce whose flesh and blood were incorporated into the mixture. The Akkadian verb for this mixing is balalu. In a previous investigation, it was established that balalu is the same root used to describe the cosmological mixing of Apsu and Tiamat's waters that produced the first gods, and is used in contexts describing the combination of distinct substances into a unified state. The third component is the Etemmu, we-ila's non-physical residuum, his ghost. Not destroyed in the killing, not sent to the underworld, transferred, bound into every human made through the procedure. And here the text does something that should not be possible to ignore. The Akkadian contains a deliberate wordplay. The god who was slaughtered, We, was the one who had Ṭēmu, intelligence, reason, the rational faculty. From his Ṭēmu and his Etemmu, his reason and his ghost, the human was made. The scribes built the word for what we are out of the word for thought and the word for the persisting dead. That pun is not decoration, it is the definition. Now read those three components against the genetic description of what you are. The physical substrate, the base genome of anatomically modern Homo sapiens, is the clay component, the material from which the form was made. The genome that is unambiguously Homo sapiens, carrying all the traits that define the species. The introgressed sequences, the Neanderthal and Denisovan and ghost population DNA, are the biological contribution from the prior workforce. Material from beings who no longer exist as distinct populations, incorporated into the substrate through contact and reproduction. Integrated, mixed in. The Akkadian verb balalu, used in the creation account and traced as the same root used for the cosmological mixing of Apsu and Tiamat, is the word for exactly this: two previously separate substances combining into a unified state. Balalu. The word for introgression, used 3,000 years before the technical term was formalized by a Swedish geneticist in the 21st century. The Etemmu component is the third layer, not physically detectable, not a sequence that leaves traces in a genome. The ghost. The genetics can identify the physical components: Homo sapiens base, Neanderthal sequences, Denisovan sequences, uncharacterized archaic sequences from ghost populations. It can measure them, map them, identify their functional effects. What genetics cannot detect is the non-physical component. By definition, a sequencing machine reads nucleotides. It does not read the category the Sumerian scribes described as transferred and integrated, but not physical. That component sits outside the instrument's measurement range. This is not a gap in the scientific method. It is the boundary of what the method was designed to measure. The genetics describes everything it can reach. The tablet describes the layer beneath that boundary. Ghost population. Eṭemmu. Not the same word, the same concept. The investigation does not claim the tablet is a genetics paper. It claims the tablet and the genetics paper are describing the same thing in the vocabulary available to each. The laboratory identified a signal in living humans from a population that left no bones. The scribe 3,000 years earlier described an invisible component from a destroyed being that persists in all humans. Both words mean ghost.
Let the convergence sit for a moment before continuing. In 2010, the genetics laboratory described a component of the human genome that comes from beings who no longer exist as a distinct population, who left no physical remains, who are known only because their signal persists in living humans. The scientists who identified this phenomenon needed a term for it. They chose Ghost population. In approximately 1700 BCE, a scribe in Babylon, copying from a tradition older still, described a component of every human being that comes from a being who was destroyed, who no longer exists in their original form, who is known only because their residuum persists in every human made through the creation procedure. The scribes who described this phenomenon needed a term for it. They used Etemmu. Both words mean ghost. Both describe the same structure, a component from a destroyed origin that persists invisibly in the living. Neither document was written for the other. Neither tradition knew the other existed. The convergence is in the vocabulary each used for the same phenomenon, independently, across a gap no cultural transmission can explain. In the 12th investigation, 3 signs from the Mesopotamian Omen archive were identified as potentially fulfilled in the current period: the celestial anomaly, the returning water, and the archive transmitting without the scribe. Those 3 signs, according to the sequential structure they appear in, precede a 4th. The 4th sign, examined in the previous investigation, is described in the Sumerian and Akkadian sources not as an external event to be observed, but as an internal change in the population that carries the inherited component. The Etemmu of we-ila, described in the creation account as transferred and bound into humanity, is not described as active throughout the current epoch. It is described as present and dormant. An integrated component that exists in every human, but does not operate until specific conditions are met. But the genetics establishes the precondition. The ghost population component in the human genome, the uncharacterized archaic sequences that geneticists cannot attribute to any identified species, is a functional part of the genome. It is not junk. Genetic sequences that persist across hundreds of thousands of years of selection pressure are generally not neutral passengers. They tend to be retained because they do something. What those sequences do in their active state, and when they become active, is currently unknown to the genetics field. The sequences are present. Their full functional role, beyond their established effects on immunity and metabolism, has not been characterized. The creation account describes the Ṭēmu component as something with a specific function, not a passive inheritance, but an active capacity. And recall the wordplay. The component is bound to Ṭēmu, the rational faculty. We-ila was specifically selected for the sacrifice because he was the one who possessed Ṭēmu, understanding. That quality was the ingredient. The Etemmu that was transferred was not generic residuum. It was the specific residuum of a being who had a cognitive capacity the creation procedure was designed to incorporate into the new workforce. What the scribes describe as dormant is not inert. It is present, integrated, and waiting. The genetics identifies the physical substrate of that component. The tablet identifies its function and the conditions under which the function becomes active. The scribes described the trigger. They described the signs that precede it. They described what the population experiences as it moves from dormant to active. That description is the next text to be opened.
The Sumerians were not doing genetics. They did not have microscopes, sequencing machines, or statistical models for detecting admixture. They had a different instrument, administrative precision applied to cosmological observation over several thousand years of record-keeping. What they produced, the creation account in the Atrahasis, is a description of what humans are made of. It is not a poetic metaphor for the human condition. In the framing this series has maintained since its first investigation, Mesopotamian scribes wrote administrative documents. They used administrative vocabulary. When they described a 3-component creation procedure with specific materials and specific operations,
They were describing what they understood to be the actual composition of the human being. Whether their instrument for arriving at that description, observation, tradition, inference, something else, was adequate to the task is a question this series does not answer. What it can do is place their description alongside the modern description and note where the vocabularies converge. Two records, separated by 6,000 years and the entire history of science. One from a genetics laboratory in Leipzig, published in 2010. Populations outside sub-Saharan Africa carry genetic sequences from species that no longer exist as distinct populations. Some of those species are known from bones. Some are known only from their signal in U. Ghost populations, ghost lineages, a persistent code from destroyed origins. One from a clay tablet in Babylon, approximately 1700 BCE. Humans were created by mixing physical substrate with material from a destroyed being from the prior workforce. That being's Etemmu, non-physical, indestructible, transferable, was bound into every human made through the procedure. The laboratory describes the physical signal. The tablet describes the non-physical component the physical signal corresponds to. Neither document was written for the other. The convergence is in the vocabulary each used for the same phenomenon, independently, across a gap no cultural transmission can explain. You are not one thing. You were not made from one thing. You carry the signal of what came before, physical and non-physical, detectable and not yet detectable. The scribe who wrote the creation account was describing you, specifically. Not humanity in the abstract, not an idealized being. You, carrying the signal of we-ila's destroyed workforce, carrying the ghost of a population that exists only as a sequence in your genome, carrying a component whose function the genetics laboratory has not yet characterized. The genetics confirms the mixture. The tablet names what was mixed and why, and what it is waiting to do. Before you go, this investigation has a gap it cannot close alone. The ghost population research is moving fast, and every few months a new study identifies another uncharacterized archaic signal in some living population. If you have seen one, a study, a population, a sequence the field has not been able to source, put it in the comments. The next part of the registry may come from a paper one of you has read and I have not. And if you want to be here when the last text is opened, the one that describes what the inherited component does when it stops being dormant, and the 3 words the scribes recorded as the trigger, then you know what the archive needs. Just stay on the list. Subscribe so that when the file updates, you are one of the ones it reaches. What that inherited component is capable of, what the scribes described as happening when it transitions from dormant to active, requires one more text. A text about the trigger. 3 words. That is the next investigation.